Hi class, this is your instructor, Skylar Huff, and we're now in Chapter 8. This is photosynthesis. The rich diversity of life that covers our Earth would be impossible without photosynthesis. Almost every oxygen atom in the air we breathe was once part of a water molecule, liberated by photosynthesis. All the energy released by the burning of coal, firewood, gasoline, and natural gas, and our bodies burning of all the food we eat, directly or indirectly, has been captured from sunlight by photosynthesis. It is vitally important, then, that we understand photosynthesis. Research may enable us to improve crop yields and land use, important goals in an increasingly crowded world. In Chapter 7, you all learned about how cells extract chemical energy from food molecules and use that energy to power their activities. In this chapter, we examine photosynthesis, the process by which organisms such as the aptly named sunflowers you see pictured in your textbook capture energy from sunlight and use it to build food molecules that are rich in chemical energy. So as we go through this chapter, we'll begin with this photosynthesis overview, the discovery of the photosynthetic processes, pigments, photosystem organization, and then the light-dependent reactions, the light-independent reaction, referred to as the Calvin cycle by way of carbon fixation, and then lastly, it'll be photorespiration. And there are those beautiful-looking sunflowers. Life is powered by sunshine. The energy used by most living cells comes ultimately from the sun and is captured by plants, algae, and some bacteria through the process of photosynthesis. The diversity of life is only possible because our planet is awash in energy streaming earthward from the sun. Each day, the radiant energy that reaches Earth equals the power from about 1 million Hiroshima-sized atomic bombs. Photosynthesis captures about 1% of this huge supply of energy and uses it to provide energy that drives all life. Photosynthesis occurs in a wide variety of organisms, and it comes in different forms. These include a form of photosynthesis that does not produce oxygen, referred to as being anoxygenic, and a form that does, oxygenic. Anoxygenic photosynthesis is found in 4 different bacterial groups: purple bacteria, green sulfur bacteria, green non-sulfur bacteria, and heliobacteria. Oxygenic photosynthesis is found in cyanobacteria, 7 groups of algae, and essentially all land plants. These 2 types of photosynthesis share similarities in the types of pigments they use to trap light energy, but they differ in their arrangement and the action of these pigments. In these plants, photosynthesis takes place primarily in the leaves. If you look at figure 8.1, it illustrates how leaves are organized. As you learned in Chapter 4, the cells of plant leaves contain organelles called chloroplasts, which are required for photosynthesis. No other structure in a plant is able to carry out photosynthesis. Photosynthesis involves 2 interrelated sets of chemical reactions. Reactions that use energy in sunlight to make ATP and reduce the electron carrier NADP+ to NADPH. This also oxidizes water to produce electrons and produces oxygen. And then secondly, reactions that use ATP and NADPH to power the synthesis of organic molecules from CO2 in the air. The first stage requires light, so the reactions involved are commonly called the light-dependent reactions. The second stage, the forming of organic molecules from CO2, is called carbon fixation. This process takes place via a cyclic series of reactions. As long as ATP and NADPH are available, The carbon fixation reactions can occur in the presence or absence of light, and so these reactions are also called the light-independent reactions. So if you look here, here is the equation for photosynthesis. This is something that you all should know. The following is a simple equation summarizing the overall process of photosynthesis. So you have carbon dioxide plus water, so that's 6 CO2 plus 12 H2O plus light, which yield glucose, which is C6H12O6 plus 6 water molecules plus 6 molecules of oxygen. That is photosynthesis. You may notice this equation is the reverse of the reaction for respiration. In respiration, glucose is oxidized to carbon dioxide using oxygen as an electron acceptor. In photosynthesis, CO2 is reduced to glucose using electrons gained from the oxidation of water. The oxidation of water and the reduction of CO2 requires energy that is provided by light. Although this statement is an oversimplification, it provides a useful global perspective. In Chapter 7, you saw that the mitochondrion's complex structure of internal and external membranes is critical to its function. The same is true for the structure and function of the chloroplast, which can also synthesize ATP by a chemiosmotic mechanism. The internal membrane of chloroplasts, called the thylakoid membrane, is a continuous phospholipid bilayer organized into flattened sacs called thylakoid discs. These are stacked in columns called grana, and this forms 3 compartments: the thylakoid membrane itself, the spaces inside and outside this membrane, and then you have the thylakoid membrane containing the enzymatic machinery to make ATP and chlorophyll and other photosynthetic pigments that capture light energy. The compartment outside of the thylakoid membrane system is called the stroma. So the stroma is that compartment outside of that thylakoid membrane. It is analogous to the matrix in the mitochondria. The stroma is a semi-liquid substance containing enzymes necessary to incorporate CO2 into organic compounds using the energy from ATP coupled with the reduction by NADPH. In the thylakoid membrane, photosynthetic pigments are organized into photosystems that absorb light, which excites an electron that can be passed to an electron carrier. Each pigment molecule within the photosystem is capable of capturing photons, which are packets of light energy. When light of a proper wavelength strikes the pigment molecule in the photosystem, the resulting excitation passes from one pigment to another. This is similar to kinetic energy being transferred along a row of upright dominoes. If you push over the first one, it falls against the next, which falls against the next, and the next, until all the dominoes have fallen over. The energy arrives at the key chlorophyll molecule in contact with the membrane-bound protein that can accept an electron. The energy is transferred as an excited electron to that protein, which passes it on to a series of other membrane proteins that put the energy to work making ATP and NADPH. These compounds are then used to build organic molecules. The photosystem thus acts as a large antenna, gathering light energy harvested by many individual pigment molecules. So that has been the thylakoid membrane. Humans have always been curious about how plants can grow and add mass without eating. The actual scientific study of this goes back 300 years and is worth considering before we look at the current view of the process of photosynthesis. The first clue came when a Belgian doctor named Jan-Baptiste van Helmont thought of a simple way to test the idea that plants grow only from materials obtained from the soil. He planted a small willow tree in a pot of soil after first weighing the tree in the soil. The tree grew in the pot for several years while Wilhelmut provided only water. At the end of 5 years, the tree had increased in size by about 74.4 kilograms. However, the soil in the pot weighed only 57 grams, less than it had 5 years earlier. This showed that the plant's increase in mass was not only produced from the soil, although Van Helmont incorrectly attributed this to the added water. 100 years passed before the story without progress until the English scientist Joseph Priestley put a living sprig of mint into air which a wax candle had burnt out. 10 days later, Priestley found that another candle could be burned in the same air. Somehow the vegetation restored the ability of the air to support combustion. Priestley went further to show that while a mouse could not breathe candle-exhausted air, air restored by vegetation was not all inconvenient to a mouse. This implies that the vegetation had added something back to the air that is vital to animals. How does vegetation restore air? That's the question. 25 years later, the Dutch physician Jan Ingenhousz demonstrated that the air was restored only in the presence of sunlight and only by a plant's green leaves, not by its roots. He proposed that the green parts of the plant carry out a process that uses sunlight to split carbon dioxide and into carbon and oxygen. He suggested that the oxygen was released as oxygen gas into the air, while the carbon atom combined with water to form carbohydrates. Other research refined his conclusions, and by the end of the 19th century, the overall reaction for photosynthesis was written as CO2 plus H2O plus light energy yields CH2O plus O2. It turns out, however, there's more to it than that. When researchers began to examine the process in detail in the 20th century, the role of light proved to be unexpectedly complex. At the beginning of the 20th century, the English plant physiologist F. Blackman measured the effects of different light intensities on CO2 concentrations and temperatures on photosynthesis. He found that at low light intensity, photosynthesis could be accelerated by increasing the amount of light, but not by increasing the temperature or CO2 concentration. And please see figure 8.3 for this. However, at high light intensities, an increase in temperature or CO2 concentration greatly accelerated photosynthesis, and this figure 8.3 is being shown to you all now. Blackman concluded that photosynthesis consists of an initial set of what he called light reactions that are largely independent of temperature, but depend on light, and a second set of dark reactions that seem to be independent of light, but limited by CO2. These so-called dark reactions are more properly called light independent, and they actually require products of the light reactions. The temperature effect Blackman observed leveled off at about 35 degrees Celsius. High temperatures cause the rate to decrease rapidly. He correctly concluded that enzymes must carry out the light independent reactions. In the 1930s, C.B. Van Niel worked at Hopkins Marine Station at Stanford, discovered that purple sulfur bacteria did not release oxygen during photosynthesis, and instead they converted hydrogen sulfide into globules of pure elemental sulfur that accumulate inside them. The striking parallels between Ingenhousz's and equation led Van Niel to propose that the generalized process of photosynthesis shown in your text and shown before yourselves right here. So in this equation, the substance H2A serves as an electron donor in photosynthesis performed by green plants. H2A is water, whereas the purple sulfur in bacteria H2A is hydrogen sulfide. The product, A, comes from splitting H2A. Therefore, oxygen produced during the green photosynthesis results from splitting water, not carbon dioxide. This could not be tested until much later, when the use of isotopes in biochemistry became common. Excuse me. So ATP and NADPH from light-independent reactions reduce CO2 to make sugars. So that was the final link in the chain of experimentation to show how the two parts of photosynthesis are actually connected. And Robin Hill demonstrated that Van Niel was right. Light energy could be harvested and used in a reduction reaction. Chloroplasts isolated from leaf cells were able to reduce dye and release oxygen in response to light. And later experiments showed that the electrons released from water were transferred to NADP+, and that the illuminated chloroplasts deprived of CO2 accumulate ATP. If CO2 was introduced, neither ATP or NADPH accumulated, and the CO2 is assimilated into organic molecules. These experiments show that photosynthesis occurs in plants within chloroplasts. They also show that light-dependent reactions use light energy to reduce NADP+ to manufacture ATP, and lastly, they demonstrate that ATP and NADPH from light reactions can be used in the subsequent reactions to reduce carbon dioxide, forming simple sugars. For plants to make use of the energy of sunlight, some biochemical structures must be present in chloroplasts and the thylakoids that absorb this energy. Molecules that absorb light energy in the visible range are termed pigments. We are most familiar with them as dyes that impart a certain color to clothing or other materials. The color we see is the color that we see is the color that is not absorbed. That is, it is reflected. To understand how plants use pigments to capture light energy, we must first review current knowledge about the nature of light. The wave nature of light produces an electromagnetic spectrum that differentiates light based on its wavelength. See figure 8.4. We are most familiar with the visible light range of this spectrum because we can actually see it. But visible light is only a small part of the entire spectrum. Visible light can be divided into its separate colors by use of a prism, which separates light based on wavelength. A particle of light, termed a photon, acts like a discrete bundle of energy. We use the wave concept of light to understand different colors of light and the particle nature of light to understand the energy transfers that occur during photosynthesis. Thus, we will refer to both wavelengths of light and to photons of light throughout the chapter. The energy content of a photon is inversely proportional to the wavelength of the light. Short wavelength light contains photons of higher energy than long wavelength light. And again, you can see this right before your faces with increasing energy as its wavelength in nanometer decreases. And you can see that decreasing energy as wavelength increases. X-rays, which contain a great deal of energy, have very short wavelengths, much shorter than those of visible light. A beam of light is able to remove electrons from certain molecules, creating an electrical current. This phenomenon is called the photoelectric effect. It occurs when photons transfer energy to electrons. The strength of the photoelectric effect depends on the wavelength of light. That is, short wavelengths are generally more effective than long ones in producing the photoelectric effect because they have more energy. In photosynthesis, chloroplasts are acting as photoelectric devices. They absorb sunlight and transfer the excited electrons to a carrier. As we unravel the details of this process, it will become clear how this process traps energy and uses it to synthesize organic compounds. When a photon strikes a molecule with sufficient energy, the molecule will absorb the photon, which will raise the electron to a higher energy level. Whether a photon's energy is absorbed depends on how much energy it carries, which is defined by its wavelength, and also on the chemical nature of the molecule it hits. As described back in Chapter 2, electrons occupy a discrete energy level in the orbitals around atomic nuclei. To boost an electron into a different energy level requires just the right amount of energy, just as the next rung on the ladder requires you to raise your foot to just the right distance. A specific atom, therefore, can absorb only certain photons of light, namely those that correspond with the atom's available energy levels. As a result, each molecule has a characteristic absorption spectrum, and the range and efficiency of photons is capable of absorbing. So as mentioned earlier in the section, pigments are good absorbers of light in the visible range. Organisms have evolved a variety of different pigments, but only two general types are used in green plant photosynthesis: chlorophylls and carotenoids. In some organisms, other molecules may absorb light energy. Chlorophylls absorb photons with narrow energy ranges. Two kinds of chlorophyll in plants, chlorophyll A and chlorophyll B, preferentially absorb violet, blue, and red light. Neither of these pigments absorb photons with wavelengths between about 500 and 600 nanometers. Light of these wavelengths is reflected, as shown here in the middle. That is where you find green light especially. Chlorophyll A is the main photosynthetic pigment in plants and cyanobacteria, and is the only pigment that can act directly to convert light energy into chemical energy. Chlorophyll B, acting as an accessory pigment or secondary light-absorbing pigment, complements and adds to the light absorption of chlorophyll A. Chlorophyll B has an absorption spectrum shifted toward the green wavelengths. Therefore, chlorophyll B can absorb photons that chlorophyll A cannot, greatly increasing the proportion of photons in sunlight that plants can harvest. In addition, a variety of different accessory pigments are found in plants, bacteria, and algae. So you can see here chlorophyll A is in that dark black line, and then chlorophyll B is the dotted line, and then thereafter we have the carotenoids, those accessory pigments, making up that gray line, making photosynthesis highly efficient across this electromagnetic spectrum by way of this visible light. Chlorophylls absorb photons by means of the excitation process, analogous to the photoelectric effect. These pigments contain a complex ring structure called porphyrin ring, with alternating single and double bonds. At the center of the ring is a magnesium atom. You can see this in figure 8.6. Photons excite electrons in the porphyrin ring, which then are channeled away through the alternating carbon single and double bond system. Electrons are not associated with the single atom or bond, are said to be delocalized with a single atom or bond, and they are said to be delocalized. Different side groups attached to the outside of the ring alter the absorption properties of different types of chlorophyll. The precise absorption spectrum is also influenced by the association of chlorophyll with different proteins. So you see there for chlorophyll A and chlorophyll B, how they differ. So all plants, algae, and cyanobacteria use chlorophyll A as their primary pigments. So it is reasonable to ask why these photosynthetic organisms do not use a pigment like retinol, the pigment in our eyes, which has a broad absorption spectrum that covers the range of 500 to 600 nanometers. The most likely hypothesis involves photoefficiency. Although retinol absorbs a broad range of wavelengths, it does so with relatively low efficiency. Chlorophyll, in contrast, absorbs only two narrow bands, but does so with high efficiency. Therefore, plants and most other photosynthetic organisms achieve far higher overall energy capture rates with chlorophyll than with other pigments. Carotenoids consist of carbon rings linked to chains with alternating single and double bonds. They can absorb photons with a wide range of energies, although they are not always highly efficient in transferring this energy. Carotenoids assist photosynthesis by capturing energy from light composed of wavelengths that are not efficiently absorbed by chlorophylls. Carotenoids also perform a valuable role in scavenging free radicals. The oxidation-reduction reactions that occur in chloroplasts can generate destructive free radicals. Carotenoids can act as all-purpose antioxidants to lessen the damage. Thus, carotenoids have a protective role in addition to their role as light-absorbing molecules. This protective role is not surprising because, unlike chlorophylls, carotenoids are found in many different kinds of organisms, including members of all three domains of life. You may have heard about eating carrots, which contain beta-carotene, can enhance vision. If you had a vitamin A deficiency, this would be true, as beta-carotene is a precursor of vitamin A. The oxidation of vitamin A produces retinol, the pigment used in vertebrate vision. Phycobiliproteins are accessory pigments found in cyanobacteria and some algae. These pigments contain a system of alternating double bonds, similar to those found in other pigments, and the molecules that transfer electrons too. So phycobilins can be organized to form another light harvesting complex that can absorb green light, which is reflected by chlorophyll. These complexes are probably ecologically important to cyanobacteria, helping them to exist in low light situations in oceans. In this habitat, green light remains because red and blue light has been absorbed by green algae closer to the surface. One way to say the role that pigments play in photosynthesis is to measure the correlation between the output of photosynthesis and the intensity of illumination. That is, how much photosynthesis is produced by how much light. Experiments on plants show that the output of photosynthesis increases linearly at low light intensities, but eventually becomes saturated with no further increase at high intensity light. Saturation occurs because all of the light absorbing capacity of the plants is in use. So during spring and summer, chlorophyll in leaves masks the presence of carotenoids and other accessory pigments. When cool fall temperatures cause leaves to cease manufacturing chlorophyll, the chlorophyll is no longer present to reflect green light, and the leaves reflect the yellow and orange light that the carotenoids and other pigments do not. Given the saturation observed with increasing light intensity, the next question is how many chlorophyll molecules have actually absorbed a photon? The question can be phrased this way: does saturation occur when all chlorophyll molecules have absorbed photons? Finding an answer required being able to measure both photosynthetic output on the basis of oxygen production and the number of chlorophyll molecules present. Using unicellular algae, chlorella, investigators obtained these values. Illuminating chlorella culture with pulses of light increasing intensity should increase the yield of oxygen per pulse until the system becomes saturated. Then oxygen production can be compared with the number of chlorophyll molecules present in the culture. The observed level of oxygen per chlorophyll molecule at saturation, however, turned out to be only 1 molecule of oxygen per 2,500 chlorophyll molecules. This result was very different from what was expected and led to the idea that light is absorbed not only by independent pigment molecules, but rather by clusters of chlorophyll and accessory pigment molecules. Light is absorbed by any one of the pigment molecules in the photosystem, and each pigment molecule transfers its excitation energy to a single molecule at a lower energy level than the others. So photosystems are those accessory pigment molecules where they are clustered. In chloroplasts and all but one class of photosynthetic prokaryotes, light is captured by photosystems. Each photosystem is a network of chlorophyll A molecules, accessory pigments, and associated proteins held within a protein matrix on the surface of a photosynthetic membrane. Like a magnifying glass focusing light on a precise point, A photosystem channels the excitation energy gathered by any one of its pigment molecules to a specific molecule, the reaction center of chlorophyll. This molecule then passes the energy of the photosystem as excited electrons that are to put to work driving the synthesis of ATP and organic molecules. A photosystem thus consists of two closely linked components: an antenna complex of hundreds of pigment molecules that gather photons and feed the captured light energy to the reaction center, and secondly, a reaction center consisting of one or more chlorophyll A molecules and a matrix of protein that passes excited electrons out of the photosystem. So briefly, the antenna complex is also called the light harvesting complex, which accurately describes its role. This light harvesting complex captures photons from sunlight and channels them to the reaction center, chlorophylls. So you're seeing right there how the antenna complex works. When the light of the proper wavelength strikes any pigment molecule within the photosystem, the light is absorbed by that pigment molecule. The excitation energy is then transferred from one molecule to another within the cluster of pigments until it encounters the reaction center of chlorophyll A. The reaction center is a transmembrane protein pigment complex. The reaction center of purple photosynthetic bacteria is simpler than the one in chloroplasts, but better understood. So briefly put, a pair of bacterial chlorophyll A molecules act as a trap for photon energy, passing the excited electron to an electron acceptor precisely positioned as its neighbor. So this is showing you when the chlorophyll in the reaction center absorbs a photon of light. 4 stages of the light-dependent reactions. As you've seen, the light-dependent reactions of photosynthesis occur in membranes. In photosynthetic bacteria, the plasma membrane itself is the photosynthetic membrane. In many bacteria, the plasma membrane folds in on itself repeatedly to produce an increased surface area. In plants and algae, photosynthesis is carried out by chloroplasts, which are thought to be the evolutionary descendants of photosynthetic bacteria. The internal thylakoid membrane is highly organized and contains the structures involved in the light-dependent reactions. For this reason, the reactions are referred to as the thylakoid reactions. The thylakoid reactions take place in 4 stages: primary photon event, charge separation, electron transport, and chemiosmosis, which I hope that you all remember from the last chapter. So at number 1, the primary photo event. A photon of light is captured by a pigment. This primary photo event excites an electron within the pigment. At charge separation, this excitation energy is transferred to the reaction center, which transfers an energetic electron to an acceptor molecule, initiating electron transport. At electron transport, which should take you all back to photorespiration, the excited electrons are shuttled along a series of electron carriers embedded within the photosystem membrane. Several of them react by transporting protons across the membrane, generating a proton gradient. Eventually, the electrons are used to reduce the final electron acceptor NADPH. Chemiosmosis. The protons that accumulate on one side of the membrane now flow back across the membrane through ATP synthase, where chemiosmotic synthesis of ATP takes place, just as it does in aerobic respiration. These 4 processes make up the 2 stages of the light-dependent reactions mentioned at the beginning of this chapter. Steps 1 through 3 represent the stage of capturing energy from light. Step 4 is the stage of producing ATP. In the rest of this section, we will discuss the evolution of photosystems and the details of photosystem function in the light-dependent reactions. Photosynthetic pigment arrays are thought to have evolved more than 2 billion years ago in bacteria, similar to the purple and green bacteria alive today. In these bacteria, a single photosystem is used that generates ATP via electron transport. This process returns the electrons back to the reaction center, and for this reason it is called a cyclic photophosphorylation. These systems do not produce oxygen, and so are also anoxygenic. So peak absorption occurs at a wavelength of 870 nanometers. So to designate this, the reaction center pigment is called P870. So in purple non-sulfur bacteria, peak absorption occurs again at 870 nanometers, and that's near infrared, not visible to human eyes. So absorption of photon by chlorophyll P870 does not raise an electron to a high enough energy level to be passed on an NADP, so they must generate reducing power in a different way. When P870 reaction center absorbs a photon, the excited electrons pass to an electron transport chain that passes the electrons back to the reaction center, generating a proton gradient for ATP synthesis. The proteins in the purple bacteria photosystem appear to be homologous to the proteins in the modern photosystem too. In green sulfur bacteria, the peak absorption occurs at 840 nanometers. Excited electrons from this photosystem can either be passed to NADPH or returned to chlorophyll by the electron transport chain, similar to that in purple bacteria. They use the electrons from hydrogen sulfide to replace those passed on to NADPH. The proteins in the green sulfur bacteria photosystem appear to be homologous to the proteins in the modern photosystem 1. In contrast to sulfur bacteria, plants have two linked photosystems. This overcomes the limitations of cyclic photophosphorylation by providing an alternative source of electrons from the oxidation of water. The oxidation of water also generates oxygen, and thus is called oxygenic photosynthesis. The non-cyclic transfer of electrons also produces NADPH, which is used in the biosynthesis of carbohydrates. One photosystem, called photosystem 1, has an absorption peak of 700 nanometers, so its reaction center pigment is called P700. This photosystem can pass electrons to NADPH, similarly to the photosystem found in sulfur bacteria discussed moments ago. The other photosystem, called photosystem 2, has a peak absorption of 680 nanometers, so its reaction center pigment is called P680. This photosystem can generate an oxidation potential high enough to oxidize water. Working together, these two photosystems carry out non-cyclic transfer of electrons that generates both ATP and NADPH. The photosystems are named 1 and 2 in the order of their discovery, not in order in which they operate in the light-dependent reactions. In plants and algae, the two photosystems are specialized for different roles in the overall process of oxygenic photosynthesis. Photosystem 1 transfers electrons ultimately to NADP+, producing NADPH. The electrons lost from photosystem 1 are replaced by the electrons from photosystem 2. So photosystem 2, with its high oxidation potential, can oxidize water to replace the electrons transferred to photosystem 1. Thus, there is an overall flow of electrons from water to NADPH. These two photosystems are connected by a complex of electron carriers called cytochrome B6F complex. This complex can use the energy from one passage of electrons to move protons across the thylakoid membrane to generate the proton gradient used by an ATP synthase enzyme. Evidence for the action of the two photosystems came from experiments that measure the rate of photosynthesis using two light beams of different wavelengths, one red and the other far red. Using both beams produce a greater than the sum of the rates using individual beams of these wavelengths. This surprising result, called the enhancement effect, can be explained by mechanisms involving two photosystems acting in a series. One photosystem absorbs preferentially in red and the other one the far red. Plants use photosystems 2 and 1 in series, first one, then the other, to produce ATP and NADPH. This two-stage process is called non-cyclic photophosphorylation. And it's called non-cyclic photophosphorylation because the path of electrons is not in a circle, so non-cyclic. The electrons ejected from the photosystems do not return to them, but rather end up at NADPH. The photosystems are replenished with electrons by splitting water. So photosystem 2 acts first. High energy electrons are generated by photosystem 2 and are used to synthesize ATP and are then passed to photosystem 1 to drive the production of NADPH, as shown here. So on average, for every pair of electrons obtained from a molecule of water, 1 molecule of NADPH and slightly more than 1 molecule of ATP is produced. So this is non-cyclic electron flow. The reaction center of photosystem 2 closely resembles the reaction center of purple bacteria. It consists of a core of 10 transmembrane protein subunits with electron transfer components and 2 P680 chlorophyll molecules arranged around this core. The light harvesting antenna complex consists of molecules of chlorophyll A and accessory pigments bound to several protein chains. The reaction center of photosystem 2 differs from the reaction center of the purple bacteria in that it also contains 4 manganese atoms. These manganese atoms are essential for the oxidation of water. Although the chemical details of oxidation of water are not entirely clear, the outline is emerging. 4 manganese atoms are bound in a cluster to reaction center proteins. 2 water molecules are then bound to this cluster of manganese atoms. When the reaction center of photosystem 2 absorbs a photon, an electron in P680 chlorophyll molecule is excited, which transfers this electron to an electron acceptor. The oxidized P680 then removes an electron from a manganese atom. The oxidized manganese atoms, with aid of the reaction center proteins, remove electrons from oxygen atoms into water molecules. This process requires the reaction center to absorb 4 photons to complete the oxidation of 2 water molecules, producing 1 oxygen in the process. So the primary electron acceptor for the light-energized electrons leaving photosystem 2 is a quinone molecule. The reduced quinone that results from accepting the pair of electrons is a strong electron donor. It passes the excited electron pair to a proton pump called B6F complex embedded within the thylakoid membrane. So the arrival of energetic electron pairs causes the BF complex pump to pump a proton into the thylakoid space. A small copper-containing protein called plastocyanin then carries the electron to photosystem 1. The reaction center of photosystem 1 consists of 1 core transmembrane complex consisting of 12 to 14 subunits with 2 bound P700 chlorophyll molecules. Energy is fed to it by an antenna complex consisting of chlorophyll A and accessory pigment molecules. Photosystem 2 accepts an electron from plastocyanin into the hole created by the exit of a light-energized electron. The absorption of a photon by photosystem 1 boosts the electron leaving the reaction center to a very high energy level. The electrons are passed to an iron sulfur protein called ferredoxin, and unlike photosystem 2 and the bacterial photosystem, the plant photosystem 1 does not rely on quinones as electron acceptors. Photosystem 1 passes electrons to ferredoxin on the stromal side of the membrane that's outside the thylakoid, and the reduced ferredoxin carries an electron with a very high potential. 2 of them from 2 molecules of ferredoxin are then donated to a molecule of NADP plus to form NADPH. The reaction is catalyzed by the membrane-bound enzyme NADP reductase. Because the reaction occurs on the stromal side of the membrane and involves the uptake of a proton forming NADPH, it contributes further to the proton gradient established during photosynthetic electron transport. And this is figure 8.15, summarizing what I just went over. Protons are pumped from the stroma into the thylakoid compartment by the B6F complex. The splitting of water also produces added protons that contribute to the gradient. The thylakoid membrane is impermeable to protons, so this creates an electrochemical gradient that can be used to synthesize ATP. The chloroplast has ATP synthase enzymes in the thylakoid membrane that form a channel allowing protons to cross back out into the stroma. These channels protrude like knobs on the external surface of the thylakoid membrane. As protons pass out of the thylakoid through the ATP synthase channel, ADP is phosphorylated to ATP and released into the stroma. And you can see that very same shown here in figure 8.15. The stroma contains enzymes that catalyze the reactions of carbon fixation, the Calvin cycle reactions. This mechanism is the same that is seen in the mitochondrial ATP synthase, and in fact, the two enzymes are evolutionarily related. This similarity of generating a proton gradient by electron transport and ATP by chemiosmosis illustrates the similarities in structure and function in mitochondria and chloroplasts. Evidence for this chemiosmotic mechanism for photophosphorylation was actually discovered earlier and formed the background for the experiments used for the mitochondrial ATP synthase. And that comes from what you would see in figure 8.16, shown here now. The passage of an electron pair from water to NADPH in the non-cyclic photophosphorylation generates one molecule of NADPH. And again, that's NADPH. And slightly more than one molecule of ATP. As you will learn later in this chapter, building organic molecules takes more energy than that. It takes 1.5 ATP molecules per NADPH molecule to fix carbon. To produce the extra ATP, many plant species are capable of short-circuiting photosystem 1, switching photosynthesis into a cyclic photophosphorylation mode, so that the light-excited electron leaving photosystem 1 is used to make ATP instead of NADPH. The energetic electrons are simply passed back to the B6F complex rather than passing on to NADPH+. The B6F complex pumps protons into the thylakoid space, adding more to the proton gradient that drives the chemiosmosis, or at least the chemiosmotic synthesis of ATP. So the relative proportions of cyclic and non-cyclic photophosphorylation in these plants determine the relative amounts of ATP and NADPH found for building organic molecules. The four complexes responsible for the light-dependent reactions, namely photosystems 1 and 2, cytochrome Bf, and ATP synthase, are not randomly arranged in the thylakoid. The thylakoid itself is no longer thought of as only stacked disks. Some models of thylakoid based on electron microscopy and other imaging depict grana as folds of interconnecting stroma lamella. This kind of arrangement is similar to the folds seen in bacterial photosynthesis that would allow more flexibility in how various complexes are arranged relative to one another. And that's how the arrangement of complexes are. Photosystem 2, cytochrome B6F, photosystem 1, and then ATP synthase. It is not random. Carbohydrates contain many carbon-hydrogen bonds that are highly reduced compared to CO2. To build carbohydrates, cells use energy and a source of electrons produced by the light-dependent reactions. Of the thylakoids. So the light-dependent reactions of the thylakoids. So for one, energy. ATP provided by cyclic and non-cyclic photophosphorylation drives the endergonic reactions. And secondly, reduction potential. NADPH provided by photosystem 1 provides a source of protons and the energetic electrons needed to bind them to the carbon atoms. Much of the light energy captured in photosynthesis ends up invested in the energy-rich carbon-hydrogen bonds of sugars. Because early research showed temperature dependence, photosynthesis was predicted to involve enzyme-catalyzed reactions. These reactions form a cycle of enzyme-catalyzed steps, much like the citric acid cycle of respiration. Unlike the citric acid cycle, however, carbon fixation is geared toward producing new compounds, so the nature of the cycle is quite different. The cycle of reactions that allow carbon fixation is called the Calvin cycle, after its discoverer, Melvin Calvin. Because the first intermediate of the cycle, phosphoglycerate, contains 3 carbon atoms, this process is called C3 photosynthesis. The key step in this process, the event that makes the reduction of CO2 possible, is the attachment of CO2 to a highly specialized organic molecule. Photosynthetic cells produce by this molecule by reassembling the bonds of 2 intermediates in glycolysis, fructose 6-phosphate and glyceraldehyde 3-phosphate, or G3P, to form energy-rich 5-carbon sugar ribulose 1,5-bisphosphate, or RUBP. CO2 reacts with RUBP to form a transient 6-carbon intermediate that immediately splits into 2 molecules of 3-carbon 3-phosphoglycerate, or PGA. That's 3-phosphoglycerate. And that's 3-phosphoglycerate, or 3-PG. This overall reaction is called carbon fixation reaction because inorganic carbon, CO2, has been incorporated into an organic form, the acid PGA. The enzyme that carries out this reaction, ribulose bisphosphate carboxylase oxygenase, usually abbreviated as Rubisco, so that's ribulose bisphosphate carboxylase oxygenase, usually abbreviated as Rubisco, is large. It's a 16 subunit enzyme found in the chloroplast stroma. We will consider how the Calvin cycle can produce 1 molecule of glucose, although this glucose is not produced directly from the cycle. If you look at figure 818, this figure being shown now. In a series of reactions, 6 molecules of CO2 are bound to 6-RUBP by Rubisco to produce 12 molecules of PGA. The 36 carbon atoms then undergo a cycle of reactions that regenerates the 6 molecules of ribulose bisphosphate, or RUBPs, used in the initial step. This leaves 2 molecules of glyceraldehyde 3-phosphate, each with 3 carbon atoms, as the net gain. You should recall that G3P as also being the product of the first half of glycolysis back in chapter 7. These 2 molecules of G3P can then be used to make 1 glucose. The net equation for the Calvin cycle is 6CO2 plus 18 ATP plus 12 NADPH plus water yields 2 glyceraldehyde 3-phosphate, 16 inorganic phosphates plus 18 ADP plus 12 NADP plus. With 6 full turns of the cycle, 6 molecules of carbon dioxide enter. 2 molecules of G3P are produced, and 6 molecules of RUBP are generated. Thus, 6 turns of the cycle produces 2 G3P that can be used to make a single glucose molecule. So the 6 turns of the cycle incorporate 6 CO2 molecules, providing enough carbon to synthesize glucose. Although 6 carbon atoms do not all end up in this molecule of glucose. The Calvin cycle can be thought of as divided into 3 phases: carbon fixation, reduction, and the regeneration of the RUBP. The carbon fixation reaction requires 2 molecules of 3-carbon acid, PGA. PGA is then reduced to G3P by reactions that are essentially a reverse part of glycolysis. And finally, the PGA is used to regenerate RUBP. 3 turns of the cycle incorporate enough carbon to produce a new molecule of G3P, and 6 turns incorporate enough carbon to synthesize 1 glucose molecule. We now know that light is required indirectly for different segments of CO2 reduction reactions. 5 5 Calvin cycle enzymes, including ribisco, are light activated. That is, they become functional and operate more efficiently in the presence of light. Light also promotes the transport of the required 3 carbon intermediates across chloroplast membranes. And finally, light promotes the influx of magnesium into the chloroplast stroma, which further activates the enzyme ribisco. Glyceraldehyde 3-phosphate is a 3-carbon sugar, a key intermediate in glycolysis. Much of it is transported out of the chloroplast to the cytoplasm of the cell, where the reversal of several reactions in glycolysis allow it to be converted into fructose 6-phosphate and glucose 1-phosphate. These products can then be used to form sucrose, a major transport sugar in plants. Sucrose is table sugar, and it is a disaccharide made of fructose and glucose. In times of intensive photosynthesis, G3P levels rise in the stroma of the chloroplast, and as a consequence, some G3P in the chloroplast is converted to glucose 1-phosphate. This takes place in a set of reactions analogous to those occurring in the cytoplasm by reversing several reactions similar to those of glycolysis. The glucose 1-phosphate is then combined into an insoluble polymer, forming long chains of starch stored as bulky starch grains in the cytoplasm. These starch grains represent stored glucose for later use. The energy-capturing metabolisms of chloroplasts studied in this chapter and mitochondria studied in Chapter 7 are intimately related if you look at Figure 8.19. Photosynthesis uses the products of respiration as starting substrates, and respiration uses the products of photosynthesis as starting substrates. The production of glucose from G3P even uses part of the ancient glycolytic pathway run in reverse. Also, the principal proteins involved in electron transport and ATP production in plants are evolutionarily related to those mitochondria. So we have discussed photosynthesis as part of cell biology because photosynthesis arose long before plants did, and because most organisms depend directly or indirectly on photosynthesis for energy that power their lives. This has been photosynthesis. Photorespiration. Evolution does not necessarily result in optimum solutions. Rather, it favors workable solutions that can be derived from features that already exist. Photosynthesis is no exception. Ribisco, the enzyme that catalyzes the key carbon-fixing reaction in photosynthesis, provides a decidedly optimal solution. This enzyme has second enzymatic activity that interferes with carbon fixation, namely that of oxidizing RUBP. In this process, called photorespiration, oxygen is incorporated into RUBP, which undergoes additional reactions to release CO2. Hence, photorespiration releases CO2, essentially undergoing carbon fixation. The carboxylation and oxidation of RUBP are catalyzed at the same time at the same active site on ribisco, and CO2 and oxygen compete with each other at this site. Under normal conditions at 25 degrees Celsius, the rate of carboxylation reaction is 4 times that of oxidation reaction, meaning that 20% of the photosynthetically fixed carbon is lost to photorespiration. This loss rises substantially as temperature increases because under hot, arid conditions, specialized openings in the leaf called stomata close to conserve water. This closing cuts off supply of CO2 entering the leaf and does not allow oxygen to exit. As a result, the low CO2 and high oxygen conditions within the leaf favor photorespiration. So this shows a stoma. And this shows how it happens, and it shows the closed stoma to the right and the open stoma to the left. And this is the figure referenced just moments ago, being figure 8.21. I think I referenced it. So these are adaptations to avoid photorespiration in just a moment. So plants that fix carbon using only C3 photosynthesis are called C3 plants. Other plants add CO2 to phosphoenolate pyruvate to form a 4-carbon molecule. This reaction is catalyzed by the enzyme PEP carboxylase. This enzyme has 2 advantages over ribisco. It has a much greater affinity for carbon than ribisco. and it does not have oxidase activity. The 4-carbon compound produced by PEP carboxylase undergoes further modification only to be eventually decarboxylated. The CO2 released by this decarboxylation is then used by rubisco in the Calvin cycle. This allows CO2 to be pumped directly to the site of rubisco, which increases the local concentration of CO2 relative to oxygen, minimizing photorespiration. The 4-carbon compound of PEP carboxylase allows CO2 to be stored in an organic form and to be released in a different cell or at a different time to keep the level of CO2 high relative to oxygen. The reduction in the yield of carbohydrate as a result of photorespiration is not trivial. C3 plants lose between 25% and 50% of their photosynthetically fixed carbon this way. The rate depends largely on temperature. In tropical climates, especially those in which the temperature is above 20 degrees Celsius, the problem is severe and has a major effect on tropical agriculture. The two main groups of plants that initially capture CO2 using PEP carboxylase differ in how they maintain high levels of CO2 relative to oxygen. In C4 plants, the capture of CO2 occurs in one cell and the decarboxylation occurs in an adjacent cell. This represents a spatial solution to the problem of photorespiration. The second group of plants, the CAM plants, perform both reactions in the same cell, but capture CO2 using PEP carboxylase at night, then decarboxylate during the day. CAM stands for crassulacean acid metabolism, after the plant family Crassulaceae, the stonecrops or hennin chicks, in which it was first discovered. This mechanism represents a temporal solution for photorespiration problem. The C4 plants. They include corn, sugarcane, sorghum, and a number of other grasses. These plants initially fix carbon using PEP carboxylase in mesophyll cells. This reaction produces the organic acid oxaloacetate, which is converted to malate and transported to bundle sheath cells that surround leaf veins. Within the bundle sheath cells, malate is decarboxylated to produce pyruvate and CO2. Because the bundle sheath cells are impermeable to CO2, the local level of CO2 is high and carbon fixation by rubisco and the Calvin cycle is efficient. The pyruvate produced by decarboxylation is transported back to the mesophyll cells, where it is converted back to PEP, then completing the cycle. The C4 pathway, although it overcomes the problem of photorespiration, does have a cost. The conversion of pyruvate back to PEP requires breaking two high-energy bonds in ATP. Thus, CO2 transported into the bundle sheath cells costs the equivalent of 2 ATP to produce a single glucose. This requires 12 additional ATP compared to the Calvin cycle alone. Despite this additional cost, C4 photosynthesis is advantageous in hot, dry climates where photorespiration would remove more than half of the carbon fixed by the usual C3 pathway alone. A second strategy to decrease photorespiration in hot regions has been adopted by the CAM plants. This includes many succulent or water-storing plants, such as the cacti, pineapples, and some members of about 24 other plant groups. In these plants, stomata open during the night and close during the day. This pattern of stomata opening and closing is the reverse of that in most plants. CAM plants initially fix CO2 using PEP carboxylase to produce oxaloacetate. The oxaloacetate is then converted into other organic acids depending on the particular CAM plant. These organic compounds accumulate during the night and are stored in the vacuole during the day. So during the day, when the stomata are closed, the organic acids are decarboxylated to yield high levels of CO2. These high levels of CO2 drive the Calvin cycle and minimize photorespiration. Like C4 plants, CAM plants use both the C3 and C4 pathways. They differ in that they use both of these pathways in the same cell. The C4 pathway at night and the C3 pathway in the day. In the C4 plants, the two pathways occur in different cells. So this has been your instructor.